My name is Rochelle Godet. I'm a professor of molecular and cellular biology at Harvard University, and my research centers on structural biology and the structure of proteins in particular. So proteins are responsible for many of the essential functions of life, and their function comes from their structure, which is determined by their sequence. So how does this linear sequence of amino acids dictate the fold or the three-dimensional arrangement of a protein? Let's start by examining the basic building blocks of proteins. Recall that a protein is a linear polymer or polypeptide of amino acids, each linked by a peptide bond. And these peptide bonds are highlighted here in yellow. There are 20 common natural amino acids that are the letters that make up most of the proteins in nature. So each amino acid has 4 different groups coming off of its central carbon, which is called the alpha carbon or C-alpha. First is just a hydrogen group. Then there's an amino group, a carboxylic acid group, and finally the R group or side chain, and that's the variable one. That's the one that varies between the 20 different common amino acids in proteins. So at the physiological pH of about 7.4, the amino group is going to be positively charged, and the carboxylic acid group is going to be a carboxylate or negatively charged. And that means that overall, the amino acid is actually going to be uncharged or neutral and a Zwitter ion because it has both a negative and a positive charge. The other important feature is that the amino acid is chiral because there are 4 different groups coming off of its C-alpha carbon. And the natural amino acids that are used by the ribosome to synthesize proteins are all L amino acids.
Do you have any tricks to remember what an L amino acid looks like?
Yes, I do. So if you take the hydrogen that's coming off of the C-alpha carbon, and then you put the nitrogen on the left and the carboxylate on the right, as you would if you're reading an amino acid sequence from N-terminus to C-terminus, then the side chain should be pointing up and towards you, like it is in this aspartic acid. To speak the language of proteins, it's important to first learn its alphabet, and that's the amino acids. The 20 common natural amino acids are shown here. There are many ways of grouping the amino acids. This arrangement, which is shown by polarity and charge, is one of these groupings. Classifying amino acids by polarity is important because their polarity affects which non-covalent interactions they can form. And these interactions are largely what gives proteins their shape. There are 3 main kinds of non-covalent interactions. The weakest ones are van der Waals interactions, such as this one between an aliphatic isoleucine and an aliphatic leucine side chain. As illustrated on the energy diagram on the right, van der Waals interactions are weak and act only over short distances, although they are present between any pair of atoms in close proximity. The distance at which the energy is minimal represents the van der Waals radius that's illustrated here by the transparent spheres. The strongest non-covalent interactions are salt bridges between pairs of charged ions. Here, a lysine side chain is paired to the C-terminal carboxylate of the protein. Depending on the polarity of the environment, a salt bridge can provide more than 10 times the binding energy of a van der Waals interaction. Finally, hydrogen bonds are 2 to 5 times stronger than van der Waals interactions, but they only occur between polar groups with permanent dipoles. And one of these polar groups is acting as a hydrogen bond donor, and the other one is a hydrogen acceptor. Here you can see a hydrogen bond within the backbone of a protein within an alpha helix. Hydrogen bonds are unique because they are directional. They are strongest when the 2 dipoles are aligned. In contrast, both the van der Waals interactions and salt bridge interactions are non-directional. So now let's survey a few of the amino acids. We won't examine them all. That's something you should do on your own. But we'll get a sampling so that we can see some of the different functionalities that each R group can have. So let's start with the 2 acidic amino acids, aspartate and glutamate. They both have a carboxylic acid group at the end of their side chain, or maybe I should say are carboxylate, because at physiological pH, they're ionized and charged, negatively charged. Now, glutamate is longer than aspartate by one methylene group. And you might think that that's not very much, that's not a big difference. But it actually makes a big difference, especially in the types of conformations or rotamers that each of these side chains can achieve. In this animation, you can see that the glutamate has many conformations that it can achieve, many more than the aspartate side chain. And that means that it might be better able to position itself exactly in the right position to interact with a substrate or a ligand in the active site of an enzyme. So although the glutamate can optimally position itself, that can come at an entropic cost. And that's because the conformational flexibility of the many rotamers will then be limited once it reaches its bound conformation, reducing its entropy. So now let's turn to histidine, which is another interesting amino acid that's often found in the active sites of enzymes. Now, histidine has a pKa for the imidazole group of its side chain of about 6, which means that it can either be uncharged or charged at physiological pH, depending on its environment. So on the left is a deprotonated or uncharged form of the histidine, whereas on the right is a protonated and positively charged form of the histidine. Now, in the neutral state, the proton can actually be on either nitrogen atom of the imidazole group, and that's illustrated here by the two alternative structures, one of them on the extreme left and one on the right of the screen. These two neutral states have different hydrogen bonding properties, as suggested by the red and blue arrows here. The transitions between the different states can be used to shuttle protons in active sites. So from histidine, we saw that the pKa's of the protein groups reflect their chemical properties. Several amino acids have polar groups that have pKa's spanning a wide range of values, as shown here. As a food for thought, consider why would tyrosine be so much more acidic than threonine and serine, even though they're all alcohols? So most amino acids are formed of carbon, nitrogen, oxygen, and hydrogen. But two of them have a sulfur atom. The first one is methionine, which is shown here. And methionine is essentially a hydrophobic residue, and it's very similar in size and shape to leucine, shown here. The second one is cysteine, which has a sulfhydryl group. And this group is interesting because it is actually quite reactive under physiological conditions. One of the reactions that it can undertake is to be oxidized to form a disulfide bond. So cysteines can react to form these disulfide bonds under oxidizing conditions, and those are conditions that are often found on the extracellular side, whereas inside cells, conditions tend to be more on the reducing side, which means that the cysteines will be found in the reduced free form. So I told you that amino acids that are used by the ribosome, the natural amino acids, are chiral and L amino acids. Well, there are actually two exceptions to this. One is glycine. Glycine has a hydrogen as a side chain, which means that it now has two hydrogens coming off of its alpha carbon, so it's not chiral. But also the small side chain means that it has fewer conformational restrictions, and that's going to be important in the process of protein folding. The second exception is proline, and proline is a cyclic amino acid, and that's because its side chain is actually covalently linked to its amino group. Now, this linkage, this covalent linkage, means that proline is actually more conformationally restricted than most amino acid. And again, this interesting conformational property comes into play when we think about protein folding. So now that we've looked at some of the amino acids in detail, let's revisit how we can classify the amino acids. This Venn diagram shows some of the many ways to classify amino acids. For example, if we look at lysine, it is charged at physiological pH because its side chain amino group carries a positive charge. It can also readily form hydrogen bonds, and therefore it's also polar.
Why is it also classified as nonpolar?
Good question. But take a look at the structure again, and I'll give you a hint. Now, aside from the charged amino group at the end of its side chain, the rest of the side chain is aliphatic or nonpolar. So that means that lysine can sometimes act as nonpolar in certain situations. So the sequence of amino acids that make up a protein is called its primary structure. And there are 4 levels of structure that make up the full 3-dimensional structure of proteins.